Greetings everyone, my name is Shoibalra and today we are back again with the continuation of the series Principles and Inheritance of Variation. Okay, so we left off by telling you, or we left off in the phase that we have done about, or we know about what are genes, what are alleles, what are the two basic laws of Mendelian inheritance, Mendel himself, and we will still continue about the video about monohybrid and dihybrid crosses. Okay, so before we start on that, okay, first I would like to bring your attention to what is a monohybrid. What is a monohybrid? Okay, what is a monohybrid? As we have discussed in the previous class as well, monohybrid is nothing but when we consider a single trait. Now we have already discussed about what are traits, what are characters. Okay, so I hope nobody has any doubt. Okay, when we consider a single trait, that cross, okay, why? Because genetics itself is the inheritance or the study of inheritance, and inheritance can only be achieved when we talk about any kind of reproduction, whether it is an asexual reproduction or whether it is a sexual reproduction. In both the cases we see that there are some inheritance patterns. Okay, when we consider just a single trait, for example, let's take plant height. Okay, now plant height itself is a single character and therefore it has a single trait. Okay, whether we consider it to be tall or whether we consider it to be dwarf, but we will consider it to be, or there is a correction, let us to avoid any confusion, right, single character. Okay, when we consider a single character such as plant height. Okay, now plant height itself will contain whether it is a tall plant, that will become its trait, or whether it is a dwarf plant, it again will become its trait. Okay, now based on this, when we make crosses, okay, we try to use mathematics to probabilistically find, or mostly how to say, in a very perchance way, we find out what is the ratio or what is the possible outcome that we can have from such a cross. Okay, so to simplify it, we will show it. Okay, let's take the example of plant height itself and we take two parents. Okay, first will be our female. Okay, and let us assume that both of these parents are true breeding types, that means they are homozygous in nature. Okay, so as we have discussed about homozygous and heterozygous in previous classes also, I will simply give you a recap. Homozygous simply means that these plants or these progenies or these genotypes, okay, or these organisms itself must contain same gametes when in meiosis. Now what does this mean? That these will have the same type of gamete. For example, if we take X and X, this is the complement of a female. Okay, this is called as a homozygous. Why? Because it contains both X and X. Okay, similarly, what will become of heterozygous? What will become of heterozygous? Heterozygous simply will be our X and Y. Why? Because this is going to create two different types of gametes. Okay, and the result itself will be a bit different. Always in meiosis we take the terms of heterozygous or a homozygous. Okay, so with this we will simply move forward. Okay, so our female will be a tall plant, but she will be a homozygous, correct? And in the same case, when we consider This is simply a recap. Okay, we have already discussed. We will be discussing about dihybrid and other expected ratios. Okay, so this is our male, and our male will be the contrary. It will be a dwarf. This is our tall, and this is our dwarf. Fine. When we make a cross, do remember this is how we proceed. That is, they will be creating their gametes, and gametes are represented like this. There is a certain notation on how to write and how to make the crosses. Okay, so to simplify it, first we will be going with our Punnett square. Okay, what is this Punnett square? We will simply keep the gametes here. Okay, this is for our F1, that is hybrids. Okay, for the first generation, what we will get is for the first generation, out of all the possible combinations, we will get as our heterozygous. This is heterozygous. This is heterozygous tall. Why? Because this capital T is dominant, or this is simply just to represent. Okay, this is controlled by the gene T, but these are called as alleles. Okay, so this capital T itself, or the capital part that we have represented, simply represents the dominant allele, or simply represents the dominant nature of this allele, which is going to suppress the expression of the smaller one, and thus we get a tall. Okay, but this is a heterozygous one. Then moving further, okay, moving further, what do we get? What happens when we cross an F1 with another F1? What happens when we do this? We get F2. That is, F2 is obtained by selfing. Okay, F2 is obtained by selfing. We got 4 progenies, right? In the first cross, we got 4 progenies. What happens when we cross those 4, or out of those 4, if we cross those 2 progenies, what is the result that we get? We get the second generation, that is F2, which is already, as I have mentioned, will be available to you only when you self it. F2 is called as, or F2 is itself called as segregating population. From here itself, it starts with the segregating populations. Okay, now what does these segregating populations mean? It simply means when we start to self, okay, we will successively go on. Okay, what happens when we self F2s? We get F3. What happens when we self F3s? We get F4s. Okay, and so on and so forth till Fn. Okay, if you are to go for higher studies, this is how you will be remembering these. What? That if we simply keep on selfing, this is how we get the various populations, and these populations are called as segregating populations. Okay, just a brief reminder, or just a brief intro. Okay, moving on. Okay, what was our purpose? We are simply going to cross again an F1, which is our, and with another F1, which is again our, correct, and both of these are heterozygous dominant tall. Okay, since dominant trait for our condition has been assumed to be tall. Okay, so this will always result in tall plants. Okay, but what happens when we cross it? We will simply make a Punnett square again. Okay, We will simply do it in the male condition. What will be the case? What will be the gametes? You will be getting these two gametes. Okay, see, this is during gamete formation. Okay, during gamete formation, if you see there are two different types of gametes, then that is an indication that this is a heterozygous organism. Okay, so for male we will simply get this. Similarly for female also we will get this. Okay, and in this when we make crosses, okay, okay, now we get something different, that is we get one capital T, two capital T small t, and only one again small t. Okay, now this first capital T, two small t, and one two capital T small t and only one small t, this is the result of a segregation. Okay, this is the result of a segregation. Now what is this segregation? As we have discussed earlier also, segregation is simply the separation. Okay, and this is what has been achieved by proving Mendel's law, that is law of segregation, that they must divide equally during gamete formation. Okay, had they not divided equally, this ratio will not be possible. Okay, and this ratio itself 1 to 1, okay, this is called as your genotypic ratio. Why? Because we are seeing it. Okay, why? Because we are seeing that we get one homozygous dominant, two heterozygous dominant, and one homozygous recessive. This is the genotypic ratio. Okay, 1 to 1 for a monohybrid cross. Okay, but what happens when we actually see, that means observe it in the physical world? What do we see? We see that this as well as this gives you the same result, that is this gives you tall, right? This gives you tall. This is also tall, this is also tall, correct? Now what happens when we get only only three talls and one short? Why? Because this is short, right? This itself is called as your phenotypic ratio, something that you observe, something that is present. Okay, something that is present. This is our phenotypic ratio, that is 3:1, correct? This is 3:1. We got it? Fine. Now to show it a bit more mathematically, okay, same thing. Okay, same thing, we will simply be doing it like this. Okay, this is what we got. Okay, for once let us assume that this is 100%. Okay, this is 100% complete organism, correct? So we can simply show it by 100% in this way also. This is also 100%, correct? Both of these are complete organisms, nothing is incomplete. Okay, now in this what happens when they form gametes? As I have told from Mendel's law of segregation, that they must divide equally. That means this should be 50% and this should be 50%, correct? That means these two gametes should be formed 50% of the time. That means every chance you get, half of the time you will be getting this type, and at the other half of the time you will be getting this type. Why? Because 50% can be written simply as half. In case of probability we show it like this, right? Right? Similarly for this also, this will also be half, this will also be half. Okay, okay. Now from this itself, okay, can you tell me how many of your individuals can you get in an F2 for a monohybrid cross? That will simply be 4. Correct? You have two types. You have two different types of gametes. Correct? And in those two different types of gametes, you will be getting 4 possible combinations. Correct? Out of which you will get, this is going to match with this. This again has the chance to match with this, same with this, and again same with this. Correct? Everyone gets equal chances to mate or to fuse with the other gamete of the other parent. Correct? So in here you will get 1/4. If we match this, we get 1/4 capital T. In this one, what do we get? Again 1/4, again 1/4 capital T, small t. Correct? In this also we get 1/4 capital T, small t. Okay? And in this case also we get 1/4 small t, small t. Okay? Same ratio. Now what we do is we can simply add these two. Why? Because they are same. Okay? They are simply the same types. So we will be getting, let me show it to you. We will simply get 1/4 capital T in ratio with half. Correct? 1/4 small t. Correct? This is the ratio that we will be getting through probability itself. Okay. 1/4 can be simply written as 25%. This is always 50%. And this again is 25%. So that means, that simply means, why I have written percentages is that whenever you cross F1 with another F1 in a monohybrid cross, okay, in a monohybrid cross, what is it that we get? We get a chance of 25% of the time you will get capital T, 25% of the time you will get small t, and 50% of the time you will get capital T, small t. These are simply how we show it in probabilities. Okay? Now, since we have discussed that part, and I hope so you have understood a bit, let us move to a bit more complex one, that is what happens when we take two characters. We call them dihybrid process. Okay? What happens when we take two crosses? We call them, or what happens when we take two characters? We call them dihybrid crosses. Okay? Now what happens in this dihybrid crosses? Simply remember that these are crosses wherein we consider two characters at the same time. Okay? Two characters at the same time. Okay? Two characters at the same time. Okay? Now what happens when we consider two characters at the same time? Let us, for example, let's take Mendel's experiment itself with plant height and pod shape. Okay? Or let us say, if we don't want to take it and be more conventional, we can simply say it seed shape. Okay? Much more easy to know. Okay. Plant height, the character itself will have two traits, that is tall, and dwarf. Okay. Similarly, seed shape will also have two traits, that is round and wrinkled. Okay, no problem. We will be simply showing it with capital R and small r for round and wrinkled. Let me write it down so that you don't get confused. This is round, this is wrinkled. So what is our dominant trait? We have taken our dominant trait to be round and the recessive trait to be wrinkled. Okay. With this information itself, let us proceed. Okay. What happens when we cross the parents, the original parents? Okay. Let us write P1. Let us write P2. Okay. This is your female. Okay. And this is your male. Again, we will be taking a true breeding type, that is a homozygous type. So we will simply write capital T, capital R, capital R. Okay. Why? Because these two are the two different characters or two different genes represented by capital T and capital R as their alleles, dominant alleles. This parent or this female parent is tall and round. Okay. Tall and round simply means tall as in plant height and round as in its seed shape. Okay. Similarly, for this also we will be taking small t and small r. This will become dwarf and wrinkled. Okay. Dwarf and wrinkled. So these will also create gametes, correct? These will also create gametes. What will be those gametes? It will be capital T, capital R. This will also be capital T, capital R. This will also be small t, small r, and this will also be small t, small r. Okay. These are the four types of gametes that you can get. Okay. Most of these will be similar. Why? Because we have taken a homozygous plant. Okay. Or a homozygous parent. Fine. If we make the Punnett square, okay. If we make the Punnett square, it will simply be, okay. It will be capital T, capital R, capital T, capital R, capital T, capital R. Sorry. This will be small t, small r, and this will also be small t, small r. So what do we get? Capital T, capital R. Okay. Capital T, capital R, small r. This is what we will be getting in all of the crosses that we face, right? And this is our F1 of a dihybrid. Okay. Okay. Remember, are you seeing something? Can you decipher something? That is, whenever we do an F1, all of the progenies that you get, all of these are identical. Identical, correct? That is why all the F1s are homogeneous in nature. Homogeneous but heterozygous in nature. This is a very important statement. Okay. Do pay note on that. What is that? All these are genetically similar, identical. Okay. Similar, identical, it's the same thing. But all of these are heterozygous in nature. Okay. They may be homogeneous, but they are heterozygous in nature. Okay. Fine. Let us move to the F2 of a dihybrid. Okay. As I have mentioned earlier, what is an F2? It is nothing but the self progeny or the self cross of a F1. Okay. So it will simply be crossed with, again, correct? This is this, this is this. When you cross it, okay, what is it that you get? You get gametes that are 4 types. Okay, that are 4 types. Now how do we create these gametes? Just make combinations. Okay, capital T, capital R. Okay, capital T will also go with small r. Okay, similarly small t will go with capital R. and small t will go with small r. Okay, this is how you make gametes for a dihybrid cross. Okay, see, this is how you make gametes for a dihybrid cross. Similarly, you will get it for this also. Okay, same. These are the gametes that have been made from this type of a organism, that is a heterozygous dihybrid cross. Okay, these are the 4 types of gametes that you will get, and then we will simply show it with a Punnett square. Okay, this will be a long one. Okay, bear with me. Okay, okay, simply write it. Okay, this is the type that we'll be getting. Write it in the same similar fashion. Okay, I'm writing it this the same way so that there is no confusion that these are gametes. Okay, now we will simply have to make the combination, the possible combinations that will arise from this. So what are the possible combinations? These are the possible combinations that you will get from a dihybrid cross when in F2 or when in F2. Okay, looks complicated, it's actually not. Okay, what happens when you first start to decipher this? This is homozygous tall and round. Okay, this is tall and round, this is tall and round, this is tall and wrinkled, this is tall and round, this is tall and round, this is tall and wrinkled, this is tall and round, this is tall and round, this is tall and round, this is wrinkled and round, this is dwarf and round, this is dwarf and round. Okay, this is tall and round, this is again tall and wrinkled, this is dwarf and round, this is dwarf and wrinkled. Okay, these are the 16 possible combinations that you can achieve. Okay, why? Because you have 4 on this side, you have 4 on that side. Okay, 4 gametes for this and 4 gametes for that, that is 4 gametes for female, 4 gametes for male. So the number of possible combinations that you will be getting is 16. Okay, now how to decipher this? Okay, just by seeing, okay, just by seeing itself, if you can count, okay, what were our dominant traits? Our dominant traits were round and tall, right? Or tall and round, either way it's fine. Okay, if it is round and tall, okay, we simply need to find out how many round and tall we get. Okay, this is tall and round, 1, this is tall and round. Okay, this is tall and round, 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 this is also tall and round, and this is also tall and round, right? So we get 9 types. Fine, we get 9 tall and round. Okay, okay, we get 9 tall and round. Okay, now what are the next types? That is, we are going to get tall and wrinkled. Okay, we need to find tall and wrinkled. So tall and wrinkled, let me show it with a different color. This is first, tall and wrinkled. Okay, then we need to find tall and wrinkled. This is second. Okay, now this is also your tall and wrinkled. Okay, these are the 3 tall but wrinkled. Okay, we need to find now dwarf and round. Okay, this is our dwarf and round. Okay, this is also our dwarf and round. Okay, and this is also our dwarf and round. So we get another 3 dwarf and round. Okay, the last one will be just this, that is dwarf and wrinkled, only one. Okay, now if you add all this, you get 9, 3, 12, 12, 3, 15 and 1, 16. All of the 16 have been accounted for. Okay, this is how we find out the dihybrid ratio for phenotypically or phenotypic dihybrid ratio of a cross. Okay, of a F2 cross. Now what happens when we, let us say, try to find out the genotypic ratio? Okay, I will give you a simple trick. Okay, what is a dihybrid? It is simply when we consider 2 traits, or we simply consider 2 characters. Okay, so if we consider that we get 1, 2, 1 from first character, right, we will also get 1, 2, 1 from the second character. Correct? This is what we will be getting. So we can simply show it in this way also. Wait, so that it does not get confusing. Wait. Okay, wait. Right? This is how we can calculate the genotypic ratio of a dihybrid. Okay, that is simply 1, 2, 1, 2, 4, 2, 1, 2, 1. Okay, just see the red lines. Okay, when you cross 1 with 1, you will always get 1. When you cross 1 with 2, you will get 2. When you cross with 1, you will get 1. Okay, this is done. When you cross the blue ones, you will get 2, 4, 2. When you cross the green ones, you will get again 1, 2, 1. Okay, this is simply how we do it mathematically. Okay, if you want to see it in real time, you can simply count it also. Okay, I'll give you a time to write it all down. Okay, and you can simply see it also. Okay, you can simply count it also. You will find that there is the same, or that is the same ratio as I have told you. Okay, now just to give you a brief idea, okay, just to give you a brief idea, mathematically, okay, or just through possibilities. Okay, you can simply say also, if you ever forget what kind of ratios you can get. Okay, just remember that for the first you will get all types of dominants. Okay, that is in our case is tall and round. You will get one possible combination of this, right? Then you get tall and wrinkled as another possibility. Okay, then you will get dwarf and round. Why? Because we are simply mixing and matching, right? Because the characters or the traits itself will be mixed and matched, that is swapped around. Okay, then the last that you get is dwarf and wrinkle. Okay, the highest percentage that you will get is of tall and round, which is 9. Okay, out of 16. Okay, the rest you will get as 3 out of 16. But in dwarf and wrinkle you get only 1 out of 16, making it a complete set. Okay, I can show it to you mathematically as well. When you got, okay, when you got these 4 combinations, okay, you can simply write it as 1/4, 1/4, 1/4, 1/4. Similarly you will get for that also. Why? Because we have simply divided this entirety into 4 parts. Okay, that is 4 gametes that have been formed of this individual itself, right? So we have divided that same individual into 4 parts, giving us 1/4, 1/4, 1/4. Okay, and whenever we cross it with the other, by law of multiplication in probability, we will always get 1/4 into 1/4, that is 1/16. Correct? So this 16 simply represents the number of possible combinations, or in mathematics we call it possible events, number of possible combinations. Okay, we get number of possible combinations. From this itself we can find out that we should get 16. Okay, but what happens when we take it a bit further? What happens when we give you, or let me give you, what happens for this? How do you represent this? Okay, or how do you make gametes of this? What will happen? A will go with B, correct? C also, and then with D also, correct? Capital wise. Similarly, it will go with A will go with small c. So the possible number of combinations that you can get, okay, let me rub it out, okay, so that does not get confusing. So the number of possible combinations or the gametes that you can form from this is A, B, correct, A, C, correct, A, D, all capitals, correct, first part. Then we get A small b, A small c, A small d, correct. Moving on, if we move to this part, smaller part, A capital B, A small A capital C, A capital D, okay. Similarly, A small b, A small c, and A small d, right? Right? How many did you get? You got 16 types, right? 8 types. 3, 4, 5, 6, 12 types. Okay. 6. Yes, you got 12. Okay. Now instead of this, okay, what we do is, this is not how we create the gametes. Okay, this was for a dihybrid type. Okay, what we do is, if I give you like this, okay, do you know what kind of gametes can you make from this? You will first create all the possible capitals. Okay, all the possible capitals. Then we go with, let us say, all the possible smalls for A. Okay. Now why am I taking all this? Because every part must be considered when you are creating the gamete. Every unique parts must be in that gamete itself. Okay. Your gamete does not simply form with simply AB, AC, D. Okay. That is not how your gamete forms. Your gamete forms with the entire combination of your all the chromosomes. Okay. And these are all unique chromosomes, or these are all unique genes. Okay. And every gene must be present in your gametes. That is why we do it with this. Okay. And now it is simply about making a mix and match columns. Okay. So we can simply keep it at B, C, D. Okay. Okay, then we can again do it like A, B, capital C, D. Okay, then again we will simply go on about. Okay, this you can take as a homework also if you want to test how to create gametes, and if you have properly understood on how to create gametes, you can simply do it. Okay, I will give you a trick on how to know, okay, how many gametes can you make out of it. Okay, take it for an example. When you take about one, take it for the first case, that is monohybrid. Okay, in case of monohybrid you only got this, correct? So you got only two types. One gene gave you two types of gametes, correct? Two gene, that is, if we take T and R itself, gave you four types, correct? Correct? Two types gave you four. Okay, you have four. Can you tell me how many gametes can you get? You will get 16. Why? It is simply 2 to the power n. Sorry. Right. Sorry. It will not be 16. Okay, it will not be 16. When you get four types, okay, you will get 2 to the power 4. That is 32 types. 16 types. My bad. Okay, 2 to the power 4 is 16. Right? This formula will help you to figure out how many gametes you require, how many gametes do you require. 2 to the power n simply represents, and n simply represents how many different types of gametes or how many different types of genes do you have. Okay, if you have TR, suppose in this case we have A, B, C and D. These are four types, so we will simply write it as 2 to the power 4, which is 16. Okay, if you add another, then you will get 2 to the power 5, which is 32. Okay, this is the 32, that is the number of possible combinations or number of possible gametes that you can have from all this. Okay, but what happens when you take an F2 of so many numbers? Suppose we have, this is simply mathematical, we are simply playing with numbers. You can try it out yourself also, because learning should be fun. Okay, so if we have 5, These are 5 genes, A, B, C, D, E. These are 5 genes. Okay. So for these 5 genes, what will we have? We can simply say that they will have 32 types of gametes. Okay. Total number of gametes that they will form is 32. Correct? So in that simple case, what is the maximum number of possible combinations that you can receive in the F2? It will simply be 4, right? 4 to the power 5. These many number of maximum combinations you can receive in F2. Okay. In F2, what is the maximum number of or maximum types or maximum combinations that you can receive in your F2 from these 32 gametes will be 4 to the power 5. Okay, that is 16, 64, 4 128 and 128 5 4, that is 2 3 8 3 10 8 3 1 512. Yeah, you will get about 512 types. Okay, you will get 512 types. Easy, right? You don't have to actually do it, it is simply to show you, but you can try to form the gametes for this. Okay, you can try simply to test whether or not you have properly understood on making gametes, because this is very important. Okay, now without further delaying, okay, let us talk about some of the rare conditions what happens in genetics. The first that I would like to point your attention to is incomplete dominance. It is not always the case that something is able to express. Okay, sometimes there are some cases wherein it is unable to do so. Okay, the first case being incomplete dominance. What happens in incomplete dominance is that the dominant allele fails to completely express itself. Now, why does it fail to completely express itself? That is also a very simple concept. Like, for example, let me write it in the next part so that I don't have to rub it out and you don't have to see to it again. Okay, what happens in that case is that let us, for example, say we need to create in a biological system, we need to create a hormone A. Okay, you need to create a hormone A. Okay, your biological system creates a hormone A. Now this hormone A must have 50 microgram per moles of a concentration. This is simply concentration. Okay, concentration of that hormone. Okay, and this A is controlled by, this hormone A is controlled by, let us say HT. Okay, this is the gene HT. Okay, this is controlled by the gene, this is a gene. Okay, gene HT. Now as we know, this gene HT will have two types, that is capital HT and small ht. Correct? Capital HT will be the dominant one and small ht will be the recessive one. Okay, this much we know. So 50 microgram per mole concentration is required to create the hormone A by the HT gene in its dominant form. Okay, in its dominant form, that is capital HT form. Okay, but when it comes in contact or when it is in that genotype, okay, when it is in that genotype, that is the genetic constitution of the organism, what happens is that in case like this HT there is no problem. Okay, in case like this, there is no problem, you will get 50, no problem. Why? Because this is a homozygous one. This is a homozygous one, no problem, you will get it. But what happens when it is in a recessive, heterozygous condition? In this condition, you will see that it is not creating 50 micrograms per mole, but rather 25 micrograms per mole. Okay, 25 micrograms per mole, which is just half in the heterozygous condition. Okay, recessive condition will always produce nothing. Okay, recessive condition produces 0. Okay, no problem here, recessive condition produces 0. But in this case, in the heterozygous part, it is creating 25 instead of the 50. But we have studied in the previous classes also, or in the previous lecture just now itself also, that whenever in heterozygous condition, the dominant allele must express itself, correct? But it is not doing so. Why is it so? Because of this. Because of this incomplete dominance. From the name itself, it is obvious that it is incomplete in nature. It cannot fully commit to the dominance nature that it possesses. Okay, and in a very short form, okay, we will discuss what is the next part, that is codominance. Okay, see, in your books it is given with the example of Snapdragon. Okay, we will explain that also, but for now simply understand what are these incomplete dominance and what is this codominance. Okay, now codominance, from the name itself, it is that the alleles fail to establish a proper dominant recessive. Okay, I'm not writing the entire thing, please understand, dominant and recessive. Okay, fails to establish a proper dominant recessive relation, when in heterozygous, my bad, it's not heterozygous, it is homozygous condition. Now what is this is that in incomplete dominance, one of those alleles was unable to express it completely. Why? Because it lacked that. Okay, when it was present with the other part, that is when it was in homozygous condition, it was compensated. Okay, the amount was compensated, that means you got 50, the required amount. But in case of codominance, what happens is, if we take the same example, okay, if we take the same example HT, okay, if we take the same example, it is HT, HT, it is HT, HT, and the last is HT and HT. See, for recessive there is no case. Okay, it is always going to produce 0. Okay, no case in this. Okay, no problem with this. The problem lies with these two. Okay, in case of incomplete dominance, it failed to produce 50, that is why it produced 25 in case of incomplete. Okay, in case of incomplete, but in case of your codominance, this is going to create 50. Okay, no problem. Why? Because alone this HT is now able to produce 50. Okay, however, when in condition of this, that is HT and HT, okay, in this case both of them, okay, in this case both of them try to express themselves. Okay, in this case both of them try to express themselves, and in doing so, okay, okay, they receive or they reach a compensation or a compromise, not compensation, compromise, where 25 is given by this and the other 25 is given by this, even though alone they can produce 50. Do you get my point? In this case, in the homozygous part, what happens is that they are trying to compromise. Okay, they are simply trying to compromise so that the desired outcome is reached. Okay, in this case, in case of codominance, it is alone able to produce it. Okay, alone it can produce its own complement. There is no requirement of compensation. But in this case, when both of them want their dominancy to overtake the other, but neither of them have the capability to do it, okay, they reach a compromise. Okay, they reach a compromise and create another genotype. Okay, create something called as a different genotype. Okay, this is a different genotype than 50. So here hormone A will have levels to it. Okay, in both the cases, in both incomplete as well as codominance, there are 3 types. Even though this is a simple type of cross, okay, you will still get 3 different types of genotypes. You will still get 3 different types of classes of your phenotypes as well. Okay, in your example or in your book's example, it is given about incomplete dominance is given about your snapdragon. Okay, that when you cross a homozygous red with a homozygous white, eventually you will get, or in the F1 you should get red, but you get pink. Why? Because neither of them are able to express completely. Okay, since it is in a heterozygous condition, it is not able to express itself completely. Sorry for that. Yeah, sorry for that. This is how incomplete dominance works. But the case is very different in codominance. What happens in that is that they receive a compromise. Why? Because both of those alleles, okay, both the alleles are trying their best to show their dominance, okay, but neither of them has the upper hand. So what they do is, to avoid any further kind of conflict, they simply come to a compromise. This is the best example that is seen in case of your blood groups. Okay, in case of blood groups, what happens is that when you see there is a blood group that is selectively controlled by the A allele, okay, and there is a B blood group that is controlled by the B allele. Okay, when both of them are together, they do not fight for dominance, they simply give you another genotype or another blood group that is called as AB blood group. Hope I am able to make you understand with this. Okay, if you have any doubts regarding whatever I have told you, please write it down in the comments so that we can also get back to you. Okay, and I hope this lecture has been helpful to you. Thank you for listening to me. Have a nice day.